Here with Michael Deering and Damien Seay, and you guys are from Del Mar College in Texas.
Yes, sir.
So whereabouts in Texas is that?
We're in Corpus Christi, which is right along the Gulf, about 2 hours away from San Antonio.
Yeah, yeah. Oh, I like the Gulf. Did you guys get hit by the oil spill there at all?
By the time any of it got to us, it was all balled up.
Was it? Yeah, yeah.
It had solidified, so
Yeah.
it didn't affect us nearly.
Okay. And what program are you? What program are you in?
I'm in the biotechnology program.
Okay. And how about you, Michael?
The same biotechnology.
Okay. What year are you?
At the end of my sophomore year, and I'll be graduating with my associate's degree at the end of this semester.
Great. Yeah.
The same.
Same. So you guys are in the same sort of cohort or group. Okay. And you've got some interesting stuff here, I guess. You want to explain a little bit about some of the work you're doing here? These look like some pretty heavy research projects.
Sure.
But what is this? What, Gossypium
Gossypium
hirsutum? Yeah.
Yes, sir.
What is? That's a bacteria.
That's a cotton.
That's a cotton.
Yeah, it's cotton.
It's
So
cotton. Oh. Okay.
Okay, so the idea is that we want to basically be able to plant cotton seeds earlier in the year without them getting frostbitten and dying.
Okay.
So we want to take this saturated fat, low palmitic acids, fatty acid, and we want to basically control the gene that makes that, so that instead of making a saturated fat, palmitic acid, it will make more unsaturated fats, which is like olive oil or vegetable oil. It's not solid. It's not a solid. So what we want to do with that is we want to increase fluidity within the cells, and that will keep them from freezing over as easily to make them more frost tolerant. So the first thing I had to do in order to do this was get about 86 samples from this guy right here. His name's Dick Auld. He's part of the Texas Tech Plant and Soil Sciences. And I had to take these seeds and analyze them with chromatography, mass spectrometry. And
These are your gas
Yes.
chromatography spikes, or those are all identifiers, right?
yes, it identifies what kind of oils are in the seed and kind of how much is in
Okay.
there.
And Damien, you were involved in this piece too?
No, I was involved in this.
Okay,
piece.
so we'll get to you in a minute.
Okay.
Okay, good. So you identified what kind of oil is in these things, and then what'd you do? You did some cloning. You pulled a gene out and.
We pulled the gene out from
Yeah.
the genomic DNA, and we did that with a PNR topo D vector. And then,
So basically, you chop it out.
yes, sir.
Okay.
That's exactly what we did.
You stick it inside some E. coli. Is that what you do?
Yes. We stuck in some E. coli.
So you chop the E. coli DNA. You stick that little piece of DNA in there. What does it do?
Then we grew it up on some
Uh-huh.
plates, and then we.
So you're growing the bacteria on
Yes,
plates.
sir.
Yep.
And then what we did after that, after the transformation is what it's called.
Yep.
We screened them on those plates for colonies that had actually accepted this gene as part of their genome. And then we took the plasmids from those, and then we basically tore those back apart after we had gotten several thousands and millions of copies of FAT-B, which is the regulatory gene for palmitic acid. And we did this with the wild type first, so that we can compare them, and we sequenced that gene to make sure that we had the right gene and
Yeah.
everything.
Yeah.
And so what we'll do from there is we'll take some of these candidates that we figured out with the GC, and the ones that are the proper candidates with like 23% or lower of the palmitic acid, we'll compare to that wild type, which has no mutation in it, and figure out exactly where that mutation inside the gene is. We'll send that information back to this guy, Dick Auld,
Yeah.
and he has the information on how he did the mutation and what kind of conditions he had, so he'd be able to recreate those and do further experiments.
Wow, that's pretty cool. Good. So the next step would be to give him give him this information, the sequencing information you pulled off, and
under the time frame that I had, what I was able to complete was figuring out which of the seeds that he gave me were candidates for low palmitic acid, and I was able to create all the primers that they'll need to do this, and I was able to sequence the wild type so that people who take over after me will be able to confirm
Great.
the mutation.
Okay. Now, Damien, you want to talk to us a little bit about what you're doing here?
Sure. So to give a bit of background information first, there's a human disorder known as Schneider-Dorfman disorder. And what that does is that causes a lipid accumulation in cells that don't normally accumulate those lipids. Well, the plant scientists saw this as a potential gold mine for the biodiesel industry. So if we can recreate this human disorder in plants, then you'll get higher oil production, and instead of only getting the oil from the seeds, you'll also get them from the leaves and the stems. So before I got to this lab this summer, they had actually replicated that disorder into the plant. So they had a high oil yield.
What kind of plants are you using?
We were using Arabidopsis thaliana, which is a common weed. So after replicating this disorder in the plant, scientists in this lab wanted to discover how the protein that's involved with that disorder interacts inside of the plant on the molecular level. And so that's where I came in, and we just, in a nutshell, tested protein-protein interactions and how it affects root length or hypocoidal length, which is also known as the stem, and if for the plant, if this protein is required for just regular germination and seedling growth, or if it's not.
Okay. So, and how far along did you get here? What step are you at here?
Well, we completed quite a few experiments, so all the ones that you see here have been completed. However, this research is still in its infancy, so a lot of these experiments can still be worked on just by simply switching things around. For example, on this first experiment here, we were testing one protein interacting with another, and we were specifically looking at the second protein. So we could do this experiment again, but focus on that first protein alone. So there's many different parts of the experiment that we can do over in a different fashion to get new information. So there's still a lot of room for growth here.
That's interesting. You're looking at all pieces of the plant, right, including the root?
Yes.
Yeah. Wow. Pretty nice. It's like fun stuff, and
Very much so.
you don't see a lot of community college programs doing this kind of research, so it's good. Thank you very much.
Thanks.
Where did Michael go? Oh, there he is. Hey, thanks. Okay. Thank you.
Thank you very much.
Oh, what are you guys going to do when you guys kind of squeeze together now? What are you guys going to do for when you get out of school?
Well, actually, the research I did over at UNT, they offered me a job for undergraduate research, so I'll be going there. And then after that, graduate school.
Good. And how about you, Damien?
Graduate school is the ultimate goal right now, with a PhD in molecular biology.
Oh, excellent. And what are you thinking for a major?
For my bachelor's, I'm thinking biochemistry, and then for my graduate, I'm thinking molecular biology as well.
Good. Wow, that's good.
Thank you very much.
All right. Good luck, guys. Thank you.